Hello, my name is Jarra Omar and I'm a senior at Poly. My project is analyzing synapses onto somatostatin interneurons. Questions regarding sensory plasticity. How can the brain alter itself to accommodate for sensory loss? What are the underlying molecular and cellular processes behind these changes? An example of this would be you being in a completely dark room and someone randomly turning the light on. Your eyes start to hurt a bit because your brain is trying to compensate for that additional input of light. Importance: 291 million people globally are either visually or auditorily impaired. Brain plasticity isn't limited to sensory function. The research here can possibly be used to help with future research that can be linked to Alzheimer's disease and other plasticity research. It can also provide a basis for devices like hearing aids, glasses, and help further innovate them, drugs, and rehabilitation. Describe a connection between neurons. Synapses are the site of communication between neurons, and chemical synapses are primarily looked at as a baseline to describe how neurons can exhibit one of two behaviors, and because they're easy to target and quantify. This is a diagram of a chemical synapse. So here we have a synaptic vesicle, which holds neurotransmitters. And once these vesicles are open, the neurotransmitters are then released, which then binds to the postsynaptic neuron. Depending on the neurotransmitter, it will kind of affect what the postsynaptic neuron does. And I'll explain that briefly. Neuron behavior. You have excitatory neurons. It increases the likelihood of another cell reaching an action potential, where the molecules bind to postsynaptic excitatory neurons, whereas inhibitory neurons decreases the likelihood of another cell reaching an action potential. Molecules then bind to postsynaptic inhibitory neurons. This is where I explain. Excitatory synapses have these glutamate neurotransmitters, which then binds to receptors on this postsynaptic cell, causing a rapid increase of voltage within the cell or depolarization, whereas inhibitory synapses have these GABA as an example, these neurotransmitters, that then binds to the receptors on the postsynaptic neuron, causing a rapid decrease in voltage within the cell, causing hyperpolarization. And these two leads to action potentials within the cell. Taking a step back for a second, there are billions of synapses all over the brain. What happens on a larger scale with a multitude of cells and synapses? For example, in one or more sensory cortices, what happens in that same system when a sensory deprivation is involved? Two different things can happen. One being cross-modal recruitment. The spared sensory cortex takes over the deprived cortex. An example would be of the somatosensory cortex, which deals with touch, of a blind person is different than it takes over, the visual cortex when reading Braille. So the overtaking of the visual cortex by the somatosensory cortex allows for blind people to have a more acute sense of touch. This diagram goes into it a bit further. So you have tactile, touch, visual, and auditory, and you have the visual cortex. There are basic inputs for all three of them, with visual being the largest. However, once you take the visual input away, you can see that the tactile auditory cortex kind of increases the strength of input within the primary visual cortex, and you can kind of understand why the visual cortex will kind of be activated in a sense when a blind person is reading Braille. Second, compensatory plasticity describes the reorganization of synapses between the spared cortices after sensory loss. Basically, the somatosensory cortex of a blind person, if we're working with that example still, is different than the somatosensory cortex of those with sight. So to compensate for this deprivation, the brain strengthens or weakens synapses in the spared cortices. In this example, this graph demonstrates the strength of the synapses between the somatosensory and auditory cortex. And after a visual deprivation is introduced, you can see that there's a big increase for some of the strengths within the synapses, kind of, describing the reorganization of synapses, allowing for strengthening or weakening of those synapses. Looking at the primary visual cortex, this cortex deals with processing visual information. So in this picture, we have the visual cortex as highlighted in purple, and it's comprised of 6 different layers within the neocortex, and it's basically just folded layers here. The first three layers of this, however, receives input first. It can receive visual and non-visual synaptic input. Cell types found in the primary visual cortex, or V1, parabellum positive interneurons, or PV, are excitatory interneurons prominently found in layer 4. Pyramidal interneurons, another excitatory interneurons found in layer 2 and 5. Somatostatin interneurons are excitatory interneurons that are found within layer 1 through 5. Vasoactive intestinal peptide positive interneurons, or VIP, which inhibits pyramidal interneurons in layers 2 and 3. Previous experimentation in lab, however, shows that there seems to be 2 distinct populations of excitatory input onto SOM cells. Questions from the study: where does this input originate from? Is it from within the visual cortex, which can be described as intracortical, or outside of V1 thalamocortical? So it could be input from auditory, or somatosensory cortex, which is sent through the thalamus to the visual cortex. To look at the SOM cells further, there are excitatory interneurons that target dendrites of other interneurons, and they're highly interconnected with little to no connection to other SOM cells. Hypothesis. So we hypothesized to see a significant difference in the distribution of intracortical synapses and thalamocortical synapses near the cell body. How are we going to test our hypothesis? So excitatory input onto SOM cells can be tagged with antibodies, basically markers to identify our targeted synapses. And VGLU1 antibody targets intracortical synapses within the visual cortex. And VGLU2 antibody targets thalamocortical synapses outside of V1. So we don't get an exact idea of where these synapses are coming from, but we do know that it could be either from the somatosensory cortex, the auditory cortex, because it's coming from the thalamus, and that's where they kind of link at. So the methods that we kind of go through are immunohistochemistry and microscopy. So we have fixed brain samples from mice that are sectioned at 10 microns. Slides are stained with appropriate antibodies, so depending on what I want to do, VGLU1, VGLU2, or both. Highlighted synapses are imaged through an LSM 700 microscope, and they are quantified with an MRS software and analyzed with GraphPad Prism. This is a merged image of a SOM cell, which is highlighted in green, VGLU1 synapses, which are in red, and VGLU2 synapses, which are in magenta, through the IMRS. So it's like a 3D image that is stacked together. It's called a Z-stack, basically multiple pictures at different locations on the cell compressed so we can see them all in one plane, and well, in 3D, excuse me. So then this is just like a rendering video of it, so you can see like the locations on the cell, on the cell body itself, on the dendrites, and kind of get an idea of how large they are in volume and how bright or whatever like you want to see. So that's basically what I worked with in terms of like the image that I see, and how I plug them out is through the IMRS software, and we can analyze it with a GraphPad Prism. Results. So we recorded dendrites 20 microns away from the cell body. Synapse density. So through an unpaired t-test, we found that p is less than 0.0001, and synapse volume through an unpaired t-test, we found that a p is equal to roughly 0.2441, and I'll tell you what that means shortly. And I'll tell you now, actually. Interpretation. So what does that mean? In layers 2 and 3 of visual cortex, colocalization density, basically points that we identified to be a synapse, of VGLU1 and VGLU2 revealed that there were more intracortical synapses near the soma of SOM interneurons, basically meaning that there are more synapses that we found that come from within the visual cortex than from outside with the thalamocortical synapses. The volume of the synapses, however, were not significantly different, meaning that the relative strength of these synapses are roughly the same. Further work. We need to look at synapses on distal dendrites in layer 1 of V1. So basically meaning dendrites from a cell body that are a bit farther away and closer to layer 1. So layer 1 is exactly where it's going to be receiving input first within the neocortex. Also, synaptic density and volume on some cells after periods of deprivation. We were looking at uh, only with mice that hasn't been deprived at all. So it's really important to see what happens after a couple of days of deprivation. Will there be more thalamocortical cells, thalamocortical synapses, excuse me, because of compensatory plasticity? Basically, there's kind of overtaking already happening, or is it roughly the same? Is it going to be distributed differently? And that's kind of the differences we want to see, or at least try to find and see. Also start standing for specific neuron input. See how the different neurons are interacting with each other after days of deprivation. That's another further work that we can do. I'd like to acknowledge Dr. Rosen, my teacher at school, who kind of helps us throughout the entire process, time management, speaking with our mentors, finding and finishing our papers, and actually kind of help get all of this together. Also, I'd like to give a big thanks to my mentor and supervisor at Hopkins, Dr. Hae Kyung Lee and Bryce Greer. They helped me through all the protocols and understanding most of the material that I needed to understand for the papers and the PowerPoints and all the presentations that I needed to do. I just wanted to say thank you for coming out and actually listening. Well, not coming out because of Corona, but actually listening to my PowerPoint. Thank you, thank you, thank you.